Well, as Paul has said, you've got a treat with the plenary speaker now, Don Kohn from UCLA, and he's going to tell you about gene therapy using hematopoietic stem cells.
Thank you, and this is, it's really great to see all the trainees here. I've always thought that CIRM does many great things, but all the trainees that support it is really one of the best. I've had, I think, a dozen Bridges students through my lab since the program began, had a training grant back in 2005. It's really important to train the next generation because we're getting older. Okay, so I'm going to tell you about hematopoietic stem cell gene therapy, and I will definitely try and finish by 6. This is my conflict of interest. I'm going to talk about a lentiviral vector that my university owns that I'm an inventor on, and it's not my retirement plan. But I do have IP on it. So the idea for gene therapy, at least it started out, it's gotten obviously much more diverse, but the initial idea was for monogenic disorders replacing the missing gene. Very logical. Cystic fibrosis, we just put a normal CFTR gene into the pulmonary system, we could treat the disease. Turns out it's a little more complicated, and especially in vivo targets are very hard to do. And so we've focused on blood cell diseases where the target is the hematopoietic stem cell, which, as I'll show you, we can take out of the body, engineer, and put back in. And so the 3 main categories of diseases are the hemoglobinopathies, for which there are now 2 approved gene therapy products, primary immune deficiencies, and storage diseases. And so this is sort of all of gene therapy on one slide. And so the field is divided into ex vivo that I'll talk about today. And you'll notice since I know Paula was speaking before him, I added B cells onto my list. It's not always there. Or in vivo. And in vivo is obviously so much easier. If you can deliver the gene intravenously or through a catheter to a specific site, that can work very well. And so there's all kinds of in vivo gene therapies that are approved drugs, for example, into the retina, into the liver, into muscle for Duchenne's. Ex vivo, where we take the cells out, we're mainly focusing on cells that are going to proliferate extensively, like stem cells or T cells. So we really need the cells to be genetically modified permanently. And so mainly we use integrating vectors like lentiviral vectors or gene editing. And the hot new things at the gene therapy meeting this year certainly were lipid nanoparticles for in vivo delivery of editor mRNAs, for example, or virus-like particles that are sort of deconstructed viruses that can deliver editing payloads. And all of what I'm talking about today is targeted to somatic cells and not the germline. And so, we don't think we're smart enough yet, I think, to be modifying the human gene pool with what we're doing now. And so all the in vivo, you have to demonstrate that your approach does not modify the germline, and ex vivo with stem cells, for example, that's not an issue. So I'm going to talk about hematopoietic stem cells as a target for gene therapy. And so here's the crash course in hematology for the newer trainees that haven't had this yet. So we're going to talk about blood as a target organ. And so what is blood? It's a suspension of cellular elements in a protein-rich liquid plasma. And if you take a sample of blood and you spin it in a centrifuge, the red cells pellet because they've got iron in them, so they're denser. The white cells form sort of a layer that's the buffy coat, and the plasma is there. So you can see 45% by volume is red cells, and that's known as the hematocrit. And as you heard from Paula, this is our union requires we show some version of this slide at all talks. This is a sort of going left to right instead of up to down differentiation from hematopoietic stem cells to the mature blood cells shown on the right. And clinically, we get our stem cells either from bone marrow, which is becoming less and less used as a source. Umbilical cord blood can be used for stem cell transplant and if it's banked. And most commonly now, as I'll show you, we use mobilized peripheral blood. And so these cells have the properties of stem cells in that they can self-renew or they can differentiate into multiple different lineages. And so the oldest stem cell therapies around are hematopoietic stem cell transplants. Hematopoietic stem cell transplants began about late 60s for SCID, as I'll talk about, and they can cure a whole list of blood cell diseases. So for all the genetic diseases that affect blood cells, if you give the patient a new source of stem cells that have the normal gene, you can make normal cells. And so again, there's the categories I talked about. This whole alphabet zoo list of immune deficiencies, hemoglobinopathies, storage diseases, and some stem cell defects. And so clinically, as I said, the way we historically mainly got stem cells from patients was a bone marrow aspirate. So your upper left, there's a patient under drapes, needles into the back of their pelvic bone, extracting bone marrow, takes a long time, and gets moderate yields of cells. So then just to follow this one, then the cells can be taken to the laboratory, gene modified. The patient often gets chemotherapy to make space. The cells are infused intravenously. So getting the transplant is actually very easy, and the cells make their way through the artery shown on the bottom, feeding the marrow, and the cells land there and start growing. And the hard part is, in the upper right, is the medical care of the patient after you just more or less blown up a bomb next to them with the chemotherapy until they recover their blood cell production. But most commonly now we collect stem cells from peripheral blood. So the observation was made that if you give a patient GCSF, or trademark Neupogen, which is used clinically to raise people's white count, 5 days in a row of that causes stem cells to leave the marrow and go into circulation. There's another drug, plerixafor, which sort of boosts that. And so by giving that, you can collect from the blood enough stem cells to do a transplant. So it's a process called leukapheresis, where the blood, some of it slowly goes to a centrifuge, it spins a gradient, collects the stem cell density, and sends everything back to the patient. So over about 3 or 4 hours, the entire blood volume will go through that machine 2 to 4 times, collecting many stem cells. And so stem cell transplant is great, but the major barrier is immunologic. And so we always think of the transplant setting that the patient can reject the incoming cells, but in stem cell transplant we also often have the opposite, because we're taking lots of donor T cells with the stem cells. They can reject their new host, and that's graft versus host disease. So clinically, when we do transplants from one person to another, that other than the underlying disease coming back, that's kind of the main problem we have to deal with clinically until magically, in most cases, tolerance develops. But occasionally GVHD could be fatal or a long-term chronic disease unto itself. And so that's where the hypothesis for gene therapy is. It's basically an autologous stem cell therapy. So using autologous stem cells that are corrected will have the same beneficial effects on blood cell production or function without the immune complications from an allogeneic or another person's stem cells. And this really should eliminate graft versus host disease risk and reduce the need for giving the patients pre-transplant and post-transplant immune suppression, which also adds to the complication. But as it turns out, we still need to give some chemotherapy to make space for these cells that we've taken out to engraft. And that's cartooned on this slide in the USC-UCLA rivalry slide. So if the patient's marrow is full of all these Trojan footballs and you want to get stem cells engrafted, we give conditioning or chemotherapy in either none or full or reduced. And so afterwards, conceptually, the marrow looks like this. In full conditioning, you more or less deplete the marrow niche of stem cells. With no conditioning, there's very little space in the niche. And with reduced intensity conditioning, there's some space. Then we give the Bruin basketballs, and then we get the level of engraftment based on the extent of ablation from the chemotherapy. So I'll show you in our SCID patients, we don't need to get full replacement. A modest amount of gene-corrected stem cells can repopulate the immune system, so we use reduced intensity conditioning, and USC and UCLA live in harmony. And so the engineering task is that we have to genetically modify the long-term pluripotent or multipotent stem cell to have a long-term effect. Everything sort of to the right of there are transient amplifying cells and mature blood cells that have various lifespans, but the stem cells, we think they'll last a lifetime if we don't damage them during the ex vivo process. And so there are 2 major ways then to do that. Add the gene with a lentiviral vector, for example, or edit the gene with CRISPR or other editing techniques. And my lab does a lot of both, but I'll just talk about one gene addition. And then there's a marker, CD34, that's on about 1% of the cells in marrow. It's not a stem cell marker, but it marks the stem and progenitor cells. So clinically, we typically select the cells for CD34 positivity to be enriched for the stem cells. And so this is the clinical cartoon. We collect the cells from the patient, we select the CD34 cells, then we either add the gene or edit the gene. During that time, the patient gets conditioned to make space, and the cells are infused. And for viral vectors, for integrating vectors, sort of the field started with mouse retroviral vectors as shown in the top, where you clone your gene into sort of the ends of the virus that will integrate. And the problem with these is that in their long terminal repeats, they have very strong enhancers and promoters. So that was good for the early vector because they made high titers and they expressed the transgene to high levels. But those enhancers can turn on the gene next to where they land. And when we treat patient cells with these vectors, we might be treating 100 million cells. So you got 100 million different integration sites, some of which by chance are near oncogenes, and that enhancer of the virus can turn it on and lead to leukemia. And that happened in early clinical trials in the early 2000s. So we've kind of switched to the second type of vector shown at the bottom, where those enhancers are deleted and they self-inactivate during reverse transcription, so they're called SYN vectors. And then the gene of interest is run off an internal promoter. And there are several, like the one I'll talk about, elongation factor, that are fairly good promoters that don't have enhancer activity or minimal enhancer activity, so they're safer. And then the other part of the problem was how do we handle the hematopoietic stem cells to keep them happy while we have them out of the body? So this is sort of the evolution of conditions over the time that I've been in the field. And so typically now we use cytokines FLT3 ligand, thrombopoietin, c-KIT ligand, and we've gone from having nothing to stromal layer to recombinant fibronectin, and now there are some transduction enhancers like poloxamers marketed as lentiboost that work well. We've moved to serum-free conditions. We've gone from retros to lentiviral vectors. And one of the advantages of lentis for stem cells is that they can transfer the gene before the cells are actually actively dividing. So you only need to have the cells in culture for about 2 days before you either freeze them or give them back to the patient, and that preserves stem cell activity. So again, the clinical cartoon is shown here, patient upper left getting leukapheresed, cells go to the GMP lab, we isolate the cells, we transduce them with usually an overnight culture to stimulate, an overnight culture with a vector, and you're done. Typically we then cryopreserve the cell and do all the release testing to make sure they meet all the criteria for sterility, potency, purity, and identity. And if they do, then the patient can get transplanted. Or we edit the cells. And so I want to talk about the disease that we've focused on for my entire adult life, pretty much, severe combined immunodeficiency. So I mentioned how bone marrow makes bone marrow contains the stem cells that make all the blood cells. There's one special cell, and that's the T cell, because it has to go to college first. So it goes to the University of T cells, the thymus gland, where it learns VDJ 101 and how to recognize self and non-self, and only the cells that have weak reactivity against self but are able to be reactive against other antigens are released, and that's the T cell repertoire. And clinically, in fact, that takes about 3 to 4 months to start happening. And in severe combined immune deficiency, SCID, that doesn't happen. And so there's a number of different genes that can give a defect in the T cells getting to the thymus or differentiating. Without T cells, B cells don't function. Some of the defects actually affect B cell or NK cells directly. So SCID is the most severe human primary immune deficiency with absent T and B cell function. NK varies due to the defects in more than any one of 20 different genes that have been identified in patients with SCID. Now we're doing screening for SCID with newborns. So all infants born in the US are tested at birth for SCID. And so from that we have a fairly good population incidence measurement of 1 in 58,000. And SCID before there were therapies was uniformly fatal in infancy. From multiple infections, chronic diarrhea, failure to thrive. And there was one child in the 70s who was maintained in a germ-free bubble for more than a decade, so this is sometimes called bubble baby disease. And the treatment primarily is bone marrow transplant. So as I said, the first transplant successful in 1968 was for a boy with SCID. He got some bone marrow from his sister, and he's in his 50s now and still has an immune system made from her bone marrow and as well. And so this works very well if there's an HLA-matched sibling to be the donor. Most patients don't have that HLA-matched sibling donor, and therefore we either use matched unrelated from the registries like NMDP or from half-matched donors like a parent or an adult. The success rates are lower because the match isn't as close as from a brother or sister. And so the specific type of SCID that we focused on is ADA SCID. So ADA is an enzyme. Those of you who've had biochemistry, this went by real quick. I know it did for me until I had to relearn it. But in our purine metabolic pathways, we have an enzyme, ADA, that removes that amino group from adenosine or deoxyadenosine to make deoxyinosine. And if you're missing that enzyme, you have SCID. And this was observed sort of serendipitously by Eloise Giblet when she was typing patients before there was HLA typing, just who had isozymes like the patient and the family, and found that the patient was missing ADA and the siblings had half normal levels. So it turns out in the absence of ADA, lymphocytes build up high levels of DATP, which is a metabolic toxin in high levels. So that's why they get SCID. And up till about a decade ago, these are the results of transplants from a donor. This is 5 different centers pooling their results. And this is survival being grafted after transplant. And so those at the top with a matched sibling or a matched family donor, like a cousin or something who might have the same HLAs, do fairly well. But those that have matched unrelated donors or mismatched unrelated donors or haploidentical donors did much worse. And so all of our trials enroll patients that don't have a matched sibling donor, although we think we're as good. And in fact, a more recent study released by a multi-center consortium looking at the survival of patients with SCID showed that in the more recent decades on your right, the outcome for even with a transplant from any donor is quite good. It statistically was not worse than gene therapy, although gene therapy still looked better. Editorial opinion. And so we've been working now for over a decade with the lentiviral vector shown here. So it's one of those SYN vectors where the long terminal repeats have their enhancers deleted. It has the elongation factor short promoter, just a small core promoter driving a codon-optimized ADA cDNA and this WPRA element that stabilizes the message. And so between those two little oops, not on there. Let's see, am I smart as Paula? Can I go backwards? Yes, okay. These two little tweaks of codon optimizing the cDNA and adding this WPR element, we get almost 10 times more protein per copy than with wild type. So even 1 copy per cell makes normal levels, in fact, excess, slightly elevated levels of ADA. And so and it was developed by our colleagues Adrian Thrasher and Bobby Gaspar in London. And so, we heard the wonderful talk by Dr. Anderson earlier where she talked about it takes a long time to get to the clinic. So this was in 2008. Bobby Gaspar and Adrian Thrasher from London and I were at a meeting. We said, we should make a lenti for ADA. That was 2008. We got into the clinic in 2013. So this is the slow road to lenti, which is a pun. And it just in red are regulatory reviews, in green are funding applications. And so it took a long time to identify the vector, do all the preclinical work, do all the applications, and ultimately submit an IND to the FDA and open trials. And lots of funding along the way from NIH, from CIRM. And so when we opened the trial, this was the clinical schema for the patients. We were giving them cells fresh from the GMP lab. So in this schema, on Monday, the patient gets admitted to the hospital. Tuesday morning, they go to the operating room and have a bone marrow harvest taken. That goes to the laboratory to start the processing. If by midnight Tuesday we know the cells are good, by the numbers, we give a single dose of busulfan chemotherapy, which is a good drug for clearing out the marrow. And then on Thursday, when we're done with the gene transfer, we take the cells from the lab to the bedside and inject them into the patient. If all goes well, a month later, we take them off their ADA enzyme that they're on, and then we follow their immune reconstitution. And we treated 21 patients under that schema. And then we moved to the cryo trial, or the frozen trial, as we called it. It's much more practical to be able to freeze the cells at the end of the gene transfer, do all the release testing before they're given to the patients. So we've sort of separated things out. So the first admission, we do the bone marrow harvest, and the patient goes home the next day. The cells are processed and then cryopreserved. Once we know they meet all the criteria, it's about 5 to 8 weeks, the patient comes back and gets treated. And now, because we don't have a rush of just a 2-day window, we can split the chemotherapy and adjust it for that patient's clearance. So we're getting much more precise chemotherapy, and then we give the cells. And we treated 12 patients under that approach. So just some pictures. Here's Nurse Lindsay bringing all the connectors we're going to need. And then in the middle is the tank bringing the frozen cells from the stem cell lab to the bedside. And then on the right is the bag of thawed cells. You can see sort of the air fluid level. It's not very translucent. There aren't a lot of cells in there, but it's enough. And then here they are going into the patient. So the syringe on your left has the cells. The syringe on your right is the saline flush. So again, you see that little bit of translucency is like 5 to 10 million per kilo CD34 cells. And so during that time, we treated patients from all over the US and Canada. This is the map, I think, to the current time of about 38 or so patients we treated. So we published the results a few years ago, pooling the results from Mattel Children's Hospital at UCLA and Great Ormond Street Hospital in London, 50 patients treated by this protocol. And what we reported was 100% survival. So every one of the patients treated is alive to the present time, and 96% event-free survival. So an event was that it didn't work and they needed to have a transplant instead. So 2 of the patients, it didn't work. Early on, the cells didn't engraft, and we never figured out why. So 2 of 5 needed a second transplant. And so this is just the survival curve. I like showing this at oncology meetings. This is the perfect survival curve. And this is T-cell numbers after transplant. So you can see over the course, again, it's a slow process, but over the course of 1 to 2 years, their T-cell numbers came up to normal. Green is the UK study that started a year before us, and blue is us in the US. And this is gene marking. And so on the left panel is the level of vector copy number, which is how many copies per cell in the granulocytes, which don't really need ADA. So they reflect how many stem cells engrafted. So it's about 0.2, or about 20% of their stem cells are engrafted. And that's enough on the right, where the T cells are in the PBMC population, to really, only those with the gene survive. So over the course, as the T cell counts are coming up, it's mainly with ones that came from the 20% that were corrected. And this is just sorting the cells and showing, again, the copy number is much higher in the lymphocytes than in the granulocytes. And we're now writing up a long-term follow-up paper, and just the results are stable. So this is survival. So we've had no deaths and no further events. And this is very small, but these are all flat curves showing T cell numbers, B cell numbers, naive T cells, et cetera. So it looks like, at least now, these patients are out 7 to 11 years, that it's stable over this time. And so that's the good news. And so we applied for many of the FDA designations that can help you get accelerated review. So this has orphan designation, breakthrough therapy, and rare pediatric disease designation. And so in 2016, in fact, this was licensed from our 2 institutions, University College London and UCLA, to a company, Orchard Therapeutics. We transferred the IND to them. We were out of the ADA business. Our kids had gone off to college. And then in 2021, Orchard announced they weren't going to continue working on it, and they returned it to us. Our kids were back in the basement. So no patient got this treatment after 2018 during that period, and we accumulated a waiting list of 30 patients with ADA SCID on enzyme waiting for treatment who didn't have MATS donors. And so, in fact, we opened a new trial at UCLA in 2013 to treat more patients. Making one major change was to switch from bone marrow to using mobilized peripheral blood. And this was kindly funded by CIRM from a grant we'd actually gotten with Orchard in 2016. There was money remaining. We were able to apply for that and get that to treat more patients. And so this is just showing the numbers of the cell doses the patient got, the 12 that got the frozen bone marrow, and now the 6 that got mobilized peripheral blood. So we're getting 2 to 3 times more cells per patient using mobilized peripheral blood. So we think that's a nice improvement. And this is just early results of the T cell counts from the 4 patients that were treated last year, showing their T cell counts are coming up nicely. So it still works. And so what I've learned, I started off doing science, but it's really drug development. And it's not a drug unless it's approved by the FDA, because you can't get reimbursed for it. And so we're trying to get FDA approval. We've been working about this. We don't have the answer, but we have a plan. And here's our plan. And one of the other things I learned from CIRM, obviously, is Gantt charts. And so our plan was in 2020 23 to open the trial, treat 3 patients, and then start working with a commercial drug manufacturing organization. To get a biological license application approved by FDA, you need to prove that you already can make the drug consistently at high commercial quality. So we needed a professional organization rather than my 3 postdocs in the factory building at UCLA to make the vector and make the cells. We then applied, we had a meeting with FDA where we talked about our plans to go to commercialization. We got their feedback. Next step was apply to CIRM for a grant to start doing this, which we did in January. CIRM then put the grants on hold, so we had to wait a little bit. We've resubmitted that grant just the end of last month. And so the plan is that we will treat 3, in the grant, the plan is to treat 3 patients next year, compassionate basis, but more importantly, 3 patients the following year with the commercial material. And in our FDA meeting, they agreed that if we can show that those 3 patients treated with commercial material meet biomarkers at 6 months for T-cell counts, gene marking, et cetera, they would accept that as a comparability. And so now once we are funded, we would work with these CDMO to develop commercial-grade vector, commercial-grade cell product. We've already collected data for a historical control set that the FDA requires you to have, and then we would treat 3 patients with the commercial product, and then we'll submit the BLA. So we're targeting that for 2 years from now. And so we're working on a path to BLA for marketing authorization. The bad news is the estimates of what it will cost for the commercial manufacturing organization to establish these capabilities are $25 to $40 million. And so we've actually spun off a company from within UCLA to handle all this. And these are just some of our former patients as they go through life. We get pictures for birthdays and other life events. And then this is my last slide before the acknowledgement, just to say that this approach of lentiviral vectors and hematopoietic stem cells has been successful for at least 18 genes that I know of, at least in early clinical trials. So multiple diseases. So even though editing is great and sexy, this approach still, lentiviral vector, I think is very valid for many diseases. And so then this is my lab group at the end of last semester at UCLA. This is our clinical trial team. These are our collaborators, and these are our sponsors for this work and other work. And you'll notice on my slide, CIRM appears 5 times, I think. Thank you. It was fast, but I know you're all hungry. I guess they're very hungry. Yes.
Thank you so much, Don, for this excellent talk. Could you comment on accessibility for the patients and how this could be in the future covered by insurances? Do you have any plans?
Yes. Well, so ADA-SCID has an advantage in that the enzyme therapy that patients get waiting for transplant costs about $300,000 a year. And so that's less than the cost of gene therapy at an academic center. I don't know, commercial pricing is another thing. And so, I think for that disease, one can make a very clear economic argument that gene therapy is going to be much cheaper. It's going to pay for itself relative to enzyme therapy. It's a problem across the rare disease field. It's a huge problem. There are many therapies that all of you and all the other smart people are developing, and they cost a lot to make because they're complicated products. And we have to find a way to get them paid for. And maybe it's a role for the government to pay for, because they are expensive to manufacture. Plus, there may be a profit motive that adds to it. But even just the cost to make them are expensive. And ones for blood cell diseases that you're doing in the context of a bone marrow transplant, that's about $250,000 just for the clinical costs for the hospitalization. So. It's a major problem that no one has a clear solution to, but I think we have to try to find a way to make them available.
Thank you very much.
Paula.
So of all the steps in the process, I think the one that is still a bit icky is the fact that you have to treat with busulfan to do even reduced intensity conditioning, because it is a chemotherapy and it has toxicity. So can you talk a bit about what might replace that in the future?
Yes, I mean, you're absolutely right. That's when we do informed consent, that's the point I spend the most time on, less so than the HIV-derived vector. As you know, there are a number of approaches using monoclonal antibodies to try and make space. Irv Weissman's group started off with anti-c-KIT. There's anti-CD45. Antibody drug conjugates seem much more potent. And so I think within 5 to 10 years, we won't be using busulfan. We'll be giving 2 or 3 antibodies to clear the marrow space out. Then the alternative beyond that will be in vivo. So giving the gene in a lipid nanoparticle and a virus-like particle and an adenoviral particle and hitting the stem cells in vivo, that's not here yet. But I think that will be great when it is. It'll be much more transportable, and you won't need bone marrow transplant doctors.
Thank you for the great talk. I was wondering if you can speak a little bit on the HSC expansion part, for possible allogenic applications, even though the autologous seems to work pretty well.
You said about HSC expansion?
Yes.
Yeah, so that's, in my mind, still remains a holy grail that we haven't reached. And there have been a number of claims of expanding HSCs. I don't know of any that I think is really clinically effective at the present time. I mean, like cord bloods tend to have a small cell dose. And so if we could expand up the true stem cells, that would be great. But I think it's been difficult. And it's really, again, as we heard some of the talks earlier, it's really we don't understand the niche that well and how to recreate it in vitro to get the cells. My analogy is we start with these perfect babies, we make a lot of teenagers. And so I think it's an unsolved problem. Yes.
Hey, Don, this is going to be a non-scientific question for you. So the students in your lab, from undergrads to graduates to postdocs, have invaluable experience in drug development, clinical translation. For this audience here, can you comment where they ended up after leaving your lab?
Sure. My undergraduates most wind up in graduate school or medical school. A few of them go to companies directly, but most of them go do further education. But pretty much all of my graduate students in the last several years after they get their PhDs have gone into industry. Either they've started companies, they've gone to startup, they've gone to biotech. Very few of them, I think I have one out of my last, however many grad students had at UCLA, 20 or so. One is an assistant professor at University of Pittsburgh. I'm very proud of him. But pretty much the rest of them have gone into industry.